Welcome to our Bonito Lab Training video on quantitative polymerase chain reaction, or qPCR. I'm Julian Liber. Retrieve the needed reagents from the -20°C freezer, namely nucleus-free water, qPCR master mix, which is wrapped in foil to keep out light, and the primers targeting your gene or genes of interest. Replace the box in the freezer. Prepare your workspace in the PCR hood by wiping down the hood surfaces and pipettes with 70% ethanol. It is often helpful to use a UV lamp to clean the hood before and after use. Often you can thaw your reagents at room temperature, just don't block too much of the airflow for the hood. Vortex your reagents, then balance them in the centrifuge and spin down. I remove the foil sleeve prior to vortexing, then replace it when setting up the reactions. The master mix contains cyber green, which can degrade with extended light exposure like most fluorophores. Other formulations in qPCR assays, such as Tachman assays, are similarly sensitive to light. I often put a sample map and mix recipe on the hood sash for easy reference. Retrieve as many tubes as you need for making mixes, one for each primer set. Avoid contaminating the supply by pouring, not reaching inside. Label the tubes with unique codes to distinguish primer sets. The first component of the mix is water, the amount of which will depend on the reaction mix you use. Avoid touching the inside surfaces of the tubes. Next we will add primers. Move each tube as you take from it to help remember that it was added. The volume needed here was just above 10 microliters, so I took twice from the same tube with new tips each time to have a higher resolution. Repeat for additional primer sets. Add the master mix next. I used Power Cyber Green, which is a 2x solution and thus will be half the final volume of the reaction. It is light sensitive, but temperature stable enough to set up the reactions at room temperature. After you finish making the mixes, you will want to place the master mix, primers, and water back in the -20°C freezer. Vortex and centrifuge the reaction mixes, then place them back in the PCR hood. Clear your workspace and reset the pipettes you are no longer using. Get out a plate and carefully place it in a rack while avoiding touching the top surface. Some plate types have already been used to calibrate the qPCR machine for a background. For this plate, I will be adding 8.5 microliters of reaction mix and 1.5 microliters of template. I'm only doing the first primer mix to start. I also depress the pipette just to the first stop to avoid adding bubbles to the wells, and I believe that this gives a more consistent reaction volume. Check that all the wells have been filled by holding up the plate. I skip wells 1A and 7G on this machine because of erroneous readings. Continue until you have filled the plate as needed. I'll cover the wells briefly. It is now time to add template, in this case reverse transcription reactions. I am adding 1.5 microliters and depressing only to the first stop. Keep track of the well order and strip orientation. I prefer to use a fresh tip box to keep track of which wells have already been filled. Take your time to ensure that the right sample ends up in the correct well on the plate. Pick each tip from the corresponding position. Take from the correct well for your template, then check that there is template liquid in the tip prior to adding it to the plate. Eject your tip and repeat. It is helpful to find an arrangement of racks, the tip box, and the waste bucket to maximize efficiency and avoid contamination. Next, I will fill the wells for the second primer set. We will need to cover the wells with a transparent film designated for qPCR use. Be careful to position it to cover all the wells and avoid creases or smudges. Take your time to position it, then seal firmly by pressing down across the entire surface. The white surfaces on the sides should be used for holding onto the film when the adhesive is exposed. Vortex the plate to mix, then centrifuge with an appropriate counterbalance. You will likely need to weigh a few plates to find a good counterbalance. Remove the plate from the centrifuge gently as to not create bubbles or splashes inside the wells. Turn on the qPCR machine using the switch on the back. You will need permission to operate this machine once trained. Move the arm up, then push back the head of the machine gently. Place your plate in the wells with row A furthest from you. Remove the side tabs from the film. Seal using the roller in the drawer under the table on which the machine rests. Close the machine and push the arm down. Open the RealPlex software on the accompanying computer using the shortcut in the upper left. Acknowledge the prompts and sign in. Select the dye your master mix or probe uses. Right-click on a well position to create an unknown sample. I typically label these with primer and sample names. You can copy and paste with Ctrl-C, Ctrl-V to populate the sheet. Double-click on a sample to change its name. Once all your samples are added, you can designate replicates by clicking on one cell, holding Shift, and clicking on another cell. Use the Group button on the toolbar to match the replicates. They must have the same name. Go to PCR Program to designate the cycle. This master mix requires 10 minutes at 95C, and usually 40 cycles of 95C for 15 seconds and 60C for 1 minute. Add a melting curve to check for specific amplification by right-clicking on the end bar. When you click the green arrow to start the cycle, the software will prompt you to save your assay. If you didn't do it earlier, you will need to designate the plate used for background calibration, the sample amount, and the probe on the plate layout screen. Save your assay again and start the cycle. Fill out the log sheet with every use. After the cycle is complete, click Yes on the prompt to begin analysis. You should hopefully see amplification curves, such as these. Click on the Edit tab, then Save Results As to export the data, usually to a flash drive. Check all samples and save the results as an Excel spreadsheet. Repeat this for the melting curve data, changing the exported file name. Save your assay before exiting. Turn off the machine and remove your plate. You should also close the computer. Thank you for watching my video about qPCR, and best of luck with your reactions.